You're listening to the Assembly on Respiratory Cell and Molecular Biology podcast, brought to you by the American Thoracic Society.
Okay, so welcome, Elizabeth, to the first episode of this new and exciting podcast that I've called the Lung Biologist Podcast. So
Thank you.
Yeah, and you're obviously Elizabeth Redente, who I'm delighted to have on the show. And I'd like to begin by sort of just introducing you and saying that your lab, from what I understand, is you aim to understand a lot of the fundamental mechanisms of lung injury and repair in models of pulmonary fibrosis. And a current focus from your lab is to understand how myeloid cells, and especially the fibroblasts, coordinately orchestrate resolution of established scar, with the ultimate aim there being to try to translate these insights into novel treatment strategies for the use in lung repair and regeneration, with a specific focus on progressive fibrosing interstitial lung diseases. But before we get into the details of your work, I would love if you would start us off by just sharing with us and the listeners a little bit more about your background, specifically your scientific training path, and sort of the road that led you to study what you are doing today.
All right. Well, thank you, Chris. And thank you for the invitation to be here and to participate in this first podcast. I'm sure it will be one of many fantastic interviews that you do. So my career path has actually been pretty straightforward, and my interest in science started very early. I had some very key teachers in middle school and high school who encouraged me to think about science and participate in the science club. And that really just sparked my overall interest in biology and in cells and in the processes associated with that. I went to my undergraduate degree is from Baylor University in biology. And I had, again, a really amazing opportunity to be in a lab and to do lab work, not just washing dishes, but actually doing experiments, doing cell culture, ELISAs, and really having the opportunity to understand what being in a lab was about. At that time, my plan had been to go to medical school. But when I got into the lab, I really felt like I'd found my place. I was able to take time to think through questions. I really felt like I could be creative. I like the concept of reading different things and putting ideas together. And our ability to find new things, to make discovery, is just incredibly exciting to me and has always kind of driven my passion for doing this work. So after college, I went to get my PhD. I did that at the University of Colorado in the School of Pharmacy. I have a PhD in molecular toxicology. I picked that program very specifically because it is a broad-based program about cellular processes, how cells respond to chemicals, to drugs, how they are misbehaving during disease processes. And I thought that broad implication of that kind of degree and those kinds of the coursework and the things we were learning could be applied to any disease I wanted to think about in the future. So that was why I picked that program, why it was important to me. And my graduate work was in a lab looking at lung cancer and macrophage programming. So this was just on the cusp of when M1 and M2 macrophages were being described. We now know that is, very naive understanding of how these cells are programmed, but at the time that's with cutting edge. And so that was my exposure to the lung. And I really became fascinated with this as an organ and understanding the cells and the interactions. I have been very fortunate in terms of my mentorship in the labs I've been in, that my PIs have been just brilliant and big thinkers and creative thinkers, which has really just inspired me to feel bold about asking my scientific questions, I think. I met my postdoctoral advisor, David Riches, while in graduate school. And when I finished my degree, I just moved down the street to National Jewish Health, which is where I'm still currently at. And Dave's lab studied macrophages in fibrosis at the time. And that's why I made that transition, because I was interested in the macrophages and applying it to a new disease. And over time, we have just kind of broadened our cell populations that we're looking at. So some macrophage work still, but really delving into the world of the mesenchymal cells and the fibroblasts in particular.
That's tremendous. So when you started out your talk talking about your initial plans to go to medical school and then pivoted based off of your experiences with your early mentorship, sort of allowing you the opportunity to explore and discover new things in science, that struck a chord with me because I think our paths kind of went in the same ultimate direction, but I came from the opposite view where I thought I just wanted to be a clinician, but then later on became exposed just like you did to science and sort of fell in love with that. And also then sort of pivoted and now, am very excited about lung biology and science. So it's funny how we can all come from different walks and different sort of initial positions, but ultimately fall in love with the lung and all the biology that's left to be discovered. So on that point, I'm curious, what do you think have been some of the most like transformative steps in advancing the field of lung biology and lung injury and repair and fibrosis? Like what is the state of the field in your view, and what are some of the greatest challenges yet to overcome?
Yeah, that's a really good question. And in many ways, it's still a hard question because we know we still have a long way to go with diseases like pulmonary fibrosis. We obviously have not developed an end-all be-all cure. But I think that, utilizing all of the new technology, when we think about the advances from single cell and now spatial transcriptomics and understanding the nuances of these cellular populations has been a really big transformative advance. We still have a long way to go as, many labs are doing this, and we have to work on nomenclature and understanding which populations we're all looking at. And then really it will be bringing it back to the biology. So kind of nomenclature of ILAs and understanding these early lung abnormalities, thinking of how we can follow these patients over time and maybe catch these fibrotic processes much earlier. I think that is very exciting. And some of the work with the PET imaging that Sydney Montesi is doing and following these active areas of disease to me is incredibly exciting. And I think we'll really be able to kind of take that clinical data and backtrack it into our animal models to really understand fibrosis development in people.
Yeah, I think it's an enormous gap in our knowledge is how do we get to the end stage, which is, oftentimes the substrate that we're analyzing, right? Explants from people who have been transplanted for this horrible disease. And we rarely get access to tissue samples from people with very early disease because they never developed symptoms enough to present for us to get access to those tissue samples. And so, like you said, being able to have new technology to be able to understand the origin, the beginning of fibrosis is going to be huge for helping us better understand how this disease progresses. And go ahead.
Oh, sorry. I was going to say it's, I think that's been one of our biggest limitations in terms of our animal models is we have so much data in our animal models with bleomycin as an example of disease initiation and the development of these processes. And even with our therapeutic interventions, right, many have been done during that development phase. And so as we can get access to patients who are in this development phase to see if our animal models are actually accurate in that process too, I think will become really important.
Critically. And then also just understand what is different between the early developing stages of human disease and the animal models that we have, like you mentioned, bleomycin, right? Because when we look at the end stage lung, there's just, it's a myriad of cell types that exist that don't normally exist there. And like you alluded to before with single cell RNA sequencing, now the heterogeneity of cells has exploded. And now we just have so many different cell types that many people have provided nomenclature for. And now we're trying to chase down those cells and see how are they talking to each other in the lung and the pathologic setting to drive disease. And so going back to the initial, earlier stages, I think will be so much simpler and easier to tease apart how they begin to initiate these processes and what sustains them.
Exactly. Right.
And speaking of, sustaining pathologic biology in the lung, a lot of your work has focused on that tail end where the cells that become activated to drive scar formation somehow for reasons we don't quite clearly understand, but I think you've provided a lot of insight into, they tend to stick around longer than we would expect. And we definitely see signatures of that in the end stage lung where these scar forming fibroblasts are resistant to apoptosis and clearing, which is what we typically expect with normal wound healing. And this is what we think a critical driver for the persistence and progression of the disease is the lack of clearing of those pathologic cells. So would you you give us a little bit of a dive into your most recent work?
Yeah, absolutely. So I think you brought up a really critical point. The end stage lung in diseases like IPF is filled with these aberrant cells that aren't supposed to be there or that have accumulated in a way that we wouldn't want. So for example, these pathologic fibroblasts that are responsible for laying down this matrix. And so when I came to National Jewish and started my postdoc, it really became clear that as we were looking at these fibroblast populations and really trying to understand their response to normal apoptotic signals, that this was an aberrant process or almost like a missing process that was happening in IPF lung or IPF fibroblast. But the problem really was when we started to look in our animal models, again, bleomycin, we weren't seeing the same thing, which is the case of many of the questions that we ask when we're comparing a progressive disease to something that is resolving spontaneously.
Indeed.
So, we started thinking about the bleomycin model, trying to kind of flip it on its head. So instead of saying this is not a great model of fibrosis, which it's not a great model of progressive fibrosis, it is a really good model of severe injury with a fibrotic repair process that gets better. And if we can start to compare what's going correctly, that it's a correct response. We want repair after injury, and then we want the lung to return to normal. So if we can look at what that correct response is and start to compare it to an IPF lung where we think this correct response is no longer happening, can we start to understand the differences and maybe think about what do we need to initiate in an IPF lung to get resolution? But to answer that question in our animal models, we really needed to develop a better system where we did not have resolving disease. And so in 2010, Tim Blackwell's group published a really nice paper with repetitive injury with bleomycin, but it was a really long model system. It took 16 weeks just to initiate the disease. So we worked to...
The poor postdocs working on that model.
So my thought was, well, I wonder what the tipping point is, and can we kind of backtrack from that and make it a little bit easier, maybe less time consuming to do. And so we kind of backed off the bleodoses and ultimately ended on our strategy of a 3-dose instillation model, which was published a couple years ago. And in this model, when we do this repetitive injury, and then the mice stay fibrotic. We've looked out 6 months and out to a year now. And so it's a really great system now where we aren't racing against that spontaneous resolution clock. So we can look at these processes that are a resolution that's already going to happen. So coming back to the idea of fibroblasts and their resistance to apoptosis, we were able to start looking now at the apoptotic signature of fibroblasts in models where the disease is not resolving and could show that they are more similar to the fibroblast seen in patient fibroblasts.
Yeah. And so having learned now that that these cells are resistant to apoptosis, I think you then, and towards the end of the paper, you introduce therapeutic measures to try and combat that resistance to apoptosis. What do you think was the biggest takeaway you learned there from those experiments?
That's a really good question. So these studies were really exciting, where we looked at the intrinsic apoptotic profile or the male apoptotic proteins on these pathogenic fibroblasts. And it turns out it's different than what's on a uninvolved fibroblast or a normal fibroblast. And based on that profile, there are therapeutics currently available that would target those cells fairly specifically, not just the fibroblasts, but potentially other pathogenic cells in the lung as well. And so it was really exciting. It was really exciting for me to take something that we had kind of mapped out the pathway and the mechanism for, and then find a therapeutic option that was available that could target it. I mean, fairly specifically, I think, without hitting normal cells. Because when we talk about loss of populations, I think when we want to clear things from a pathologic lung, we don't really want to get rid of everything, because that would also be bad. We don't want to kill all of the fibroblasts or all of the epithelium, just those that are participating in the fibrotic process.
Well, that's the key point, right? That's the key point with that this drug that you're introducing is that even if it does target the fibroblast somewhat specifically, it's likely to have off-target effects on other cells. But the goal here is to clear out all the bad cells, right? The ones that don't really belong there in health. So even if it is spilling over into other subsets, it's probably only affecting those subsets that are also contributing to pathology. So you're kind of hitting, the major target, the scar-forming cells, but also, potentially as well impacting the other subsets that are contributing to this process. So that's kind of a neat way to approach it.
Yeah, it's great. And I think we've shown it really nicely targeting the fibroblasts. Other groups, David Lagares, when he was in Andy Taker's lab, was looking at the skin, fibroblasts in the skin. Brent Carter's group has looked at macrophages, and he has just completed a phase 1 trial with one of these compounds, looking at safety, which I think went very well. And so the other thing I really like about these compounds and this pathway is that it's not just our work that is showing that it's looking positive or effective. There's multiple groups across the country. So to me, that's even better because it's a reproducible result.
That's wonderful. Yeah, that's fantastic. And so do you think now with the results of your data and the data you commented on from other labs that we're moving towards maybe a new dawn of combination therapies for IPF and progressive fibrosing lung disease? Right now, the state of the art, for those who aren't aware, we only really have 2 drugs to treat these diseases, and they're almost never used in combination, kind of just use one or the other. But it'd be interesting to get your thoughts on where you think we're headed in terms of therapies.
Yeah, it's a good question. And I'll just, say I'm not a clinician, but I do think, what nintedanib and pirfenidone are targeting is not the same thing that these other compounds that are maybe manipulating the apoptotic potential of these cells are. And so while one drug maybe is slowing down collagen production, let's say, we still need to probably get rid of certain cell populations or encourage development pathways to turn back on so that there's normal repair that can occur in the lung. So I think ultimately, combination therapy is going to be what's important. I would suspect the next question will be, do you throw all the drugs in at once, or it would need to be like sequential over time as you take care of one problem, you kind of deal with the next and go forward. So I'm sure it'll be a complicated process. But we're right now doing studies like combination studies with nintedanib and these 3 mimetics to look at that exactly.
Oh, that'll be interesting to see how the combination effect is, right? Yeah, because if the cells aren't gotten rid of, you still have the same problem. So we're going to have to target it using different approaches like you mentioned, which is I think where we're headed. And so you've done a lot of work, and it's been a tremendous value to the field, I think, learning how these cells are being resistant to apoptosis. And this work was done by you at National Jewish, which is still ranked as one of the top hospitals in the world for pulmonary medicine. And so I was going to ask you, a plug for NJH. What is the research environment like there in Denver? And, how has it been suited for you to accomplish all the work that you've done so far?
Yes, that's a good question. So I think I've been incredibly lucky to have landed at National Jewish. And I've also been able to extend my career here. So I was a trainee, but have become faculty and graduated to being an independent person at the same institution, which I think isn't always easy, but it's been very supportive. And I think as well, the close collaborations we have with the University of Colorado and their outstanding pulmonary department makes being here quite ideal because we have access to a fellowship program as well as graduate students that way. But I really think what I love about National Jewish are the people and my colleagues. It is just a really positive place to work. The collaborations and interactions among the faculty are phenomenal. And in particular, I just want to highlight here, we have a really amazing cohort of early and mid-career women faculty. We also have many women who are in leadership positions, division chairs. So it's just also a place that is very welcoming and encouraging to that demographic. So even on the floor I work on, there's 4 physician scientists or PhDs. We collaborate. It's so positive and supportive. And last year, all 4 of us got RO1s. And so it's testament. I know it's super exciting. just this testament to this supportive environment of really brilliant people who want to help and work together because we know we're stronger together. So that, for me, that part.
That's great. That should really be a push sort of, global wide to get, more representation in science. And sounds like, leading the way in that in some respects, which is fantastic to hear. And hopefully we see more of that in the future, certainly. And I think a lot of the work you've done there has provided you, tremendous success, which has springboarded you into leadership at the American Thoracic Society Assembly of the Respiratory Cell Molecular Biology section. And so for listeners who may not be all that familiar with what the RCMB is or does, would you provide a little bit more information about what your role is in this assembly and what might the benefits be for those who are thinking about getting involved, both for trainees and for maybe more established PIs?
Sure. So the Respiratory Cell Molecular Biology, or RCMB, is one of the assemblies, which the assemblies are, kind of cohorts of topical groups within ATS. So it's an opportunity to kind of be aligned scientifically with your colleagues in a smaller, more manageable group. Right now, I am serving as the chair of this committee, which essentially means I participate right now in the board of ATS, so at a higher governance level, but then also, interact and help facilitate all the other subcommittees in the assembly. This includes all of the programming that happens with abstracts. It includes all the awards. It includes communication like the Web Committee that Chris is on, as early career members are getting involved. There are apprenticeship programs where you can essentially join a committee as an apprentice to learn about kind of the behind the scenes workings of how the conference gets put together or how programming is initiated. This year in particular, there has been open calls for participation at the upcoming meeting for moderators for poster discussion sessions and mini symposium, as well as the ability to participate in the early career programs like resident boot camp and new faculty boot camp. The other thing that RCMB is really involved in, which I love, is the Networking Supercenter and the Science and Innovation Center. This is outside of programming, but really highlights new and innovative science that's upcoming. There's a lot of career development sessions that happen there. They have the speakeasy where it's, kind of the big name and you can hear their stories, as well as hosting the PhD reception. RCMB really strives to have innovative programming throughout the year as well. And that's really spearheaded by our early career professional working group and our aging working group, as well as the Web Committee. And so I think it's just a really great way to kind of find when we first go to meetings, often we go with our mentors. And I remember very distinctly watching mine, interact with all these people and thinking, wow, he just knows everybody. And there's these connections. And as you become involved, you meet your people and eventually you become that same person where you go to the meeting and you now are interacting with your colleagues and scientists across the country and across the world, really. And I think this is just a really, again, supportive environment where not just the assembly, but the members really care about each other and they care about the success of our early career members. And it just provides a unique opportunity to really get to know people in the field and share your work.
Absolutely. Yeah. Like you said before, sort of a hub for latest and greatest, most innovative science is often revealed there in plenary sessions or poster discussion. There's a lot to be learned and gained there, but probably the most valuable asset, as you said, is the networking. And I remember also going to these meetings in the beginning and thinking, my PI was just born with all of these contacts from the womb, just knew everyone. And it seemed so bizarre. And I felt like I had no place there. But after, doing that over and over, you kind of get to meet more and more people and they keep going to the same meetings. You see each other over and over again. And that, I think that's a great place to foster collaboration and to also learn different perspectives about science to push it forward, which is a great aspect of the conference that I certainly look forward to every year when I go. So, a major plug for the conference for anyone who has not yet been or is considering of joining the assembly, please, enroll because the value and the benefits are enormous.
And ATS is is big. It's a big conference. It's a big society, and plugging into an assembly is a way to kind of make that more manageable as well.
Certainly. Is there anything coming up to this upcoming conference in 2025 that you think might be different, or you're looking most forward to?
And I think
More of the same greatness, probably.
it's more in the same greatness. I mean, the basic science core topic this year, which is on disease across lifespan and aging, I just think is so exciting. And so I'm really looking forward to seeing how that programming gets shaped out.
One of the nice things for people who may think the conference is so daunting, because it does include, a wide breadth of science, is that I think certainly last year, I remember you shared a PDF document that had a roadmap for all the basic science core talks and poster sessions, which I use as my sort of treasure map for the entire conference. So hopefully that will be shared again by the assembly for the next conference. We can have a clear way of navigating where all the great talks are going to be.
Yes, that is definitely the plan. So I'm glad that was helpful.
Yeah, it was tremendously helpful. So yeah, thank you again for that. And let's see. So you're talking about all the benefits that trainees can get. It'd be great to now kind of pivot a little bit and provide some, through your own introspection, maybe some advice as to the trainees who may be on the listening to the podcast. And it would be great if you could go back and think about if you could redo or restart your scientific training all over again. Would you think you would do anything differently? Or if not, what advice do you think you may have been given that allowed you to be as successful as you are today?
Great. That's a really good question. I think I have been very fortunate when I look back and think about my path and my opportunities. I probably wouldn't change the choices that I made or the path that I took. But what I have really learned is that it's not usually a linear path, and there's lots of ways to get to where we are now, to be studying science, to be studying the lung, and whether that's a physician scientist track or a PhD track, we all kind of can end up in the same place through lots of different ways. And so some of my advice is to not feel like maybe what has been considered a really standard track in the past. I mean, that's definitely what I did. College, graduate school, try to get a faculty job. It's way more flexible now, and there's way more opportunities, even outside of academic, true academics, to participate in research and science. And as industry has grown, and there are amazing companies out there that really care about the research too. And so there's a lot of ways to become involved and have this be your career. It isn't a one size fits all. So I think that that's important. I also really think that this is a challenging career in many ways. Funding, we're now, integrating research and patient care, so many things. It's almost a continual, at least for me, a continual decision to stay in this and to find the joy and be excited by the science. And so with that, I also think it's okay to recognize that maybe we've done all this work, and many of us we're very driven, and we've worked very hard to get to this point. But maybe what we want now in our 30s or 40s isn't the same that we wanted when we're 20. We want to pursue this career in a different way, maybe advocacy or thinking about policy or something different. And also, I think science, where it is now versus when I started or when our mentor started, is definitely not the same playing field. The rules have changed. It's harder. There's more expectations. We're all pulled. And so it's also, I just want to say that it's like also okay if that doesn't fit what you want anymore either. Like the world we're playing in this career is certainly changed, and it will continue to change, and that's okay. And there's way more options now. And so not always feel like you have to be stuck in a certain spot or a certain path to be successful in pulmonary or a scientific career.
Yeah, that's so true. You mentioned how the game has changed, right? In my own literature review, going back deep into the old published articles that are out there, you're looking at journal articles in the 70s, 80s, you see these papers in high tier journals that only had like a few Western blots, and that was it. That's all you needed. But now it's like you need 12 supplemental figures of like deep omics data to qualify for entry into some of these journals. And it's just a tremendous amount of work that's required. And I think really opens the door for team science and for
Yeah.
a lot more collaboration, which certainly we think is one way to establish that, certainly is at the ATS conferences, but it's just in general something to think about at places like NJH, where it sounds like a lot of collaboration is happening there, like you mentioned, and certainly hopefully more of that to come in the future to provide, more people with unique training and expertise to come together and offer what they have to really push the field forward, which is what we need to truly understand how these diseases are still, why they're still impenetrable to our drugs, right? There's something that we're missing that we haven't yet discovered. So there's a lot more to do. And so you talked about in your training, being mentored by Dave Riches, which, a phenomenal
Right.
mentor. Is there any piece of advice that was given to you by him or other mentors along the way that you think sort of shaped your approach to science?
Yeah, I mean, it's maybe been less about direct advice, but I think it's been more about like leading by example is maybe a better way that I've kind of adapted and been able to like stay positive in this career. So again, just remembering that this is for many of us a passion, but it's a job. It's a really important job, and we're contributing to patient lives and to society, but that balance is really good. So I've had mentors all throughout my career that, personal life is a priority. And so keeping that also at the forefront in addition to working really hard and making progress scientifically has been, I think, really helpful for me to not just have too much of tunnel vision as I go through my life. And that, to remember that it takes all of us a long time to get to where we are. And often we see people's lives as a Facebook or Instagram or whatever social media post. It's like a perfect little square. You only see the Nature publication or the grants that are funded, and you don't see everything behind that, all the grants that were resubmitted 3 or 4 times, or the paper rejections, and that, we're all standing on all of that hard work that isn't often acknowledged or seen. I think that's helpful for me to know too.
Yeah, absolutely. I think there's so much work that goes unpublished because of things that you mentioned, and it's hard to get a project over the finish line, and I think in many ways we do applaud a lot of the papers that do make it there because of all the work that's required. But no matter the journal, I think it's important to be kind and to be respectful and to just appreciate all the energy and time that goes into accomplishing all this work that we're trying to do to share the discovery and to learn more about lung biology. And so along the way, is there one particular chance that you took that stands out in your memory as like one that you were exceptionally glad you took that kind of helped get you to here? I think you mentioned pivoting from like the cancer biology field over into lung, using like the macrophages as an example to bridge the two fields. That might be an example, but if not that one, is there anything else, maybe even in your most recent paper, like reaching for specific drugs that you decided to use or anything else like that you think was an interesting choice?
Yeah, so I think that I could answer that question 2 ways. I think there are scientific chances you take, right, where you try to ask a new question. Certainly for me, one of those was some earlier work I was doing with my postdoc with TNF-alpha, where we were suggesting that this pro-inflammatory cytokine could actually be beneficial in the resolution of fibrosis and lung injury. And that was, a challenge to get that idea out there, but one that we, continued to pursue, and I think successfully. So I think that would be a research area where we tried to kind of push the boundaries a little bit and take a chance on a new idea. But the other thing I think I would say about taking a chance is about getting involved and trying to find my voice, because I'm a pretty introverted person and don't necessarily like being out in the spotlight. But for me, making a choice to participate in ATS and to feel like I could try for leadership opportunities and have a voice and help to shape, what's going on, that was a big reach and step for me, one that I'm glad I was able to take and follow through with.
Well, I think a lot of us listening are looking forward to the changes under your leadership at the upcoming ATS conference through the Assembly. And so I think we will leave it leave it at that and just say, Elizabeth, thank you so much for joining this first podcast episode. And it was a wonderful conversation, and look forward to hearing more about your work in the future.
All right. Thank you, Chris. I appreciate it.
Yeah.
Thank you for joining us today. To learn more, visit our website at thoracic.org. Find more podcasts from the ATS assemblies on YouTube. Don't forget to like, comment, and subscribe so you never miss a show.